Alright guys, this is Antonio Urbano. I'm doing my presentation through video form of my Mitosis and Meiosis lab for Fundamentals of Biology 1. Just going right into this. First we're going to be going over mitosis. And we're going to go over a brief overview of the cell cycle. We're going to be going over this area, mitosis. This is going to be our main focus today. But just going over G1, the G1 section of the cell cycle is when the cell just lives its life and just grows in general. And then that follows into the S section, the S part of the cell cycle, and that's when all of the chromosomes are being duplicated in the cell, which follows into G2, which is where it's just getting ready to go into mitosis. So let's hop right into mitosis, and the first phase of mitosis is going to be prophase, and that's when the chromosomes over here begin condensing into, the euchromatin begin condensing into its chromosome form, and the microtubules over here just kind of get ready for the following process. Next phase is prometaphase, and this is kind of an in-between part between prophase and metaphase, hence the name prometaphase. It's kind of a combination of the two. This is just kind of a significant part because the nucleus membrane has been broken up and the chromosomes over here have finally been condensed into their form, and the microtubules have been attached to the centromeres of the chromosomes. Alright, that brings us into metaphase. This is pretty cool because meta and all the chromosomes are lined up in the middle. So M and M, which was what we kind of went over in my lecture, but M and M, metaphase, and all the chromosomes are lined up in the middle, and all of our microtubules are attached to the centromeres, ready to be pulled apart at the point of the microtubules are attached to the centromeres and also the centrioles, which are these parts over here. And once they begin to be pulled apart, that leads us into anaphase. The stereotypical chromosome look of the X's over here has now been split apart into their each individual chromatids, and as we can see, just the cell begins splitting apart over here, and which brings us into telophase and cytokinesis. And this is when the nuclear membrane starts to reform. The cytoplasm is beginning to be split up between the cells. The cells getting pinched off by microfilaments over here, and beginning to separate into, and is being separated into two cells during this section. And the chromosomes are being turned into euchromatin, which is like that spaghetti kind of form. Anyway, now we go into meiosis, and there we go. This is going to, now we're going into meiosis. This is just a brief overview of what's going to be going on in meiosis. We have our individual haploid chromatids over here. Wait, camera person. Wait, come on. Wait, you can come closer, don't worry. Over here. And then we actually see that they begin, once they're in their stereotypical chromosome form, they pinch together at these points, which we later see that they pick up traits from the other chromosome that they paired up with. And then they come up with, but we're going to be going into that more detail following. That was just the general overview. So now we're back into meiosis, the first part of meiosis. Alright, actually, wait, camera person, can you come back up again? Actually, the spaghetti form of the chromosomes, the euchromatin, you can actually see right here. That is in spaghetti form, and it turns into chromosome form as we go into prophase. So that's actually a good way of viewing how it was previously and then how it's turned into in prophase. So that's actually pretty cool. So okay. So going into the first part of meiosis, we have prophase, where the chromosomes are condensed, as we saw, from spaghetti euchromatin form to just regular chromosome form in prophase 1, and then also the microtubules are getting ready. Just like in mitosis, we have our prophase where they get ready, metaphase where they all line up and they attach to the centromeres, and then in anaphase they begin to be separated over here, and then which leads us into the second part, which the first part ends in telophase over here, where it has become two completely separate cells, and which begins the second part with prophase 2, very brief, which goes on for a very brief moment. Metaphase, where they line up in the middle again, and we can actually see here. Actually, I'll go into that later. So, all right. Which leads us into metaphase 2, where they all line up in the middle, and anaphase 1, where they split up, and in cytokinesis, where they each have their own pair over here, and you can actually see that each pair has a different variation from the other pair. They each have their own variation, which is actually really cool. Which we can see over here. They picked up the traits by pinching, coming through. And that actually leads us into genetic diversity, which what I was going to go into. And the first area where we can get genetic diversity from is prophase 1. And I was mentioning it before. I completely forgot I had a slide on it. But we're going to be going over it. We have our two chromatids over here. And then they come over in prophase 1 and they actually pinch. And then what happens is they pick up traits. Right over here, they picked up some alleles from the blue chromatid over here. And they're now on the red one. And then the blue one now has traits from the red one in prophase 1. And this actually happens again. Well, not the same process, but another process of genetic diversity that can happen in metaphase 2. And this happens when we go over here, prophase, this is the metaphase, this is the anaphase when they begin to separate, and then we come into prophase, and then, prophase happens over here, metaphase, and then anaphase, and actually we can see in the metaphase 2 that at this point, right here, when it splits up, we have an option to have the dominant be on top or the recessive be over here. And then as we follow down that process, that brings us to, once they split apart in anaphase 2, we have 1/4 of a chance for each outcome to happen, and we have a dominant gene paired with a dominant one, or we have recessive, or if it went down the other path, we have a dominant recessive or a recessive and dominant trait. So that's actually really cool. And that leads us into our last thing, the haploid versus diploid just concept. And with haploids, we have one copy of each chromosome. Over here, they don't have a pair, they're just by themselves. And diploids have a pair. They're the same size, and the centromere is located in the same point. And I thought an easy way to visualize this was through this process right here, just through like sexual reproduction of cells, where we have one sperm and it has its haploids by itself. We have the haploids here and they're by themselves, and we have the haploids from the egg over here and by themselves, and when they combine, they form the diploids over here, the pairs of the fertilization of the egg. So yeah, that's my presentation on meiosis and mitosis, and yeah, hope you all enjoy.